And good morning, everyone. So everybody in the audience say hello.
Hello.
Pretty lame audience.
Hello.
That's fine. Well, I didn't have my band, you know. All right. So let's start to review, get back where we were. And that is the initial steps in the breakdown of glucose. And that, of course, is the pathway known as glycolysis. Glycolysis, the splitting of sugar. Glycolysis is the pathway that pretty much every organism on the planet uses. Bacteria use it, we use it, everybody uses glycolysis. So this is a very old, evolutionarily old set of biochemical pathways. So we start out with the 6-carbon glucose, and we're going to immediately add the phosphate from 2 ATPs. They're now 2 ADPs. And the glucose molecule splits into 2 3-carbon molecules. Now again, when you take biochemistry, you can memorize the name of phosphoglyceraldehyde, the structure. For our purposes, no. The structure is just the number of carbons is fine. The next step is introducing our electron carrier. We have an NAD back up to each one of these 3-carbon molecules and pick up an electron. So we finally now begin to reduce this molecule a bit, or excuse me, we're going to oxidize the glucose, and we were reducing the electron carrier. We still haven't left any carbons loose. Still have 3 carbons on each one of these guys. The next step is the ATP regenerating step, as it's called, in which 2 adenosine diphosphates can be phosphorylated into 2 adenosine triphosphates, and each 3-carbon molecule can do that. And as we'll see, this is a kind of rephosphorylation. It's actually a little bit odd, or, well, actually it's very, very primitive. It is referred to as substrate level phosphorylation. We'll talk about that in a little bit. It's also the point where if you take creatine is involved, not in exactly this way, but in a similar way in terms of substrate level phosphorylation. And so sometimes weightlifters will take creatine, or people that are doing like essentially anaerobic type exercise, not aerobic exercise. It doesn't do you any good, but you can take it. All right, so now we actually are at the end of the game with glycolysis. We still have these 2 3-carbon molecules, and let's go ahead and name them because we will use this. This is the 2 3-carbon molecules. These are known as pyruvic acid, are often referred to as pyruvate. So we've taken glucose and broken it down into 2 pyruvates. If we sort of have our tote board, which we'll use over here, we can say this: glycolysis, what goes in is the 6-carbon glucose, and we have to have 2 ATP, 2 NAD. Now, yes, you need ADP here as well, but I never put those into the tote board because, frankly, if all of your adenosines are in the triphosphate form, you don't need to be doing this. You also aren't doing any kind of work or any kind of metabolism, but that's the whole point, is that you always have lots of ADP floating around. All right, so what comes out is the 2 3-carbon pyruvate and 4 ATP and 2 NADH. So it took 2 ATP to get this thing started. You get 4 out. We think of that as a net gain of 2 ATP. So we'll just say right here then, our net gain is 2 ATP. And if you recall over here, we were promised 36. So there's a lot more energy to be extracted from this stuff. All right, so this whole reaction takes place in the cytoplasm. And our next step, so glycolysis is in the cytoplasm. The next step is to take pyruvate and place it into the mitochondria. Now again, depending on if you're a lumper or a splitter, and goodness knows I'm a lumper, this next step is actually viewed as a separate step. And it is known as the oxidation of pyruvate. So as pyruvate makes its way into the mitochondria, so here's the mitochondrial membrane, and we'll be talking a lot more about mitochondria here in a bit. As pyruvate is transiting across the mitochondrial membrane, a carbon is removed. And so we now have a carbon coming off as CO2. We're only going to take one of these 3 carbons. The next one's following right behind it or right beside it, but we're only going to look at one. What this means, though, is that we really need to count up to 3 carbons to make sure all our carbons are gone. So that's our first carbon exhaled out. Everybody take a breath out. Now, once we take a carbon from a 3-carbon molecule, this ain't real high math, we got 2 carbons. And if you recall in our nomenclature, this is the ethyl group. It's also known as the acetate or acetyl group. The acetyl group here that's left, as it goes into the mitochondria, it gets complexed up with a vitamin molecule that is known as coenzyme A. And coenzyme A is a great big heterocyclic molecule with lots of electron resonance, and it's another one of these Wikipedia philosophical things. Why is it coupled to this vitamin as it makes a transit? Because it's going to immediately be popped off. I don't know why. There's probably some biochemical pathway that makes this useful, but regardless, the molecule that goes into the next pathway is known as acetyl-CoA. So our next molecule that we're going to be chewing on is the 2-carbon acetyl-CoA. All right. So this is going into the next step. First step is glycolysis, depending on how you want to lump things. Second step is the oxidation of pyruvate. Third step now is the breakdown of the 2 carbons that are left. And just as a little historical point, very good. The glycolysis pathway was figured out first. And it is known, if you think about it, you're going from a big molecule into smaller molecules, what you expect when you're breaking something down. And so this was figured out fairly quickly, even back, I think, before the beginning of the 20th century, if not very early before World War I. So now, after World War I, there were a lot of German scientists that were disgruntled with the whole business going on in Germany. This guy, Hans Krebs, moved to the UK, and he took with him this device known as a manometer. And as I always think of it, is a very elaborate crack pipe. It is a tube with lots of blown glass wiggles and things, and you can place a piece of liver, fresh liver, in the bottom of it, put different kinds of metabolites in there, and watch very carefully the evolution of carbon dioxide as bubbles. And so you can monitor, by looking at the amount of bubbles, you can monitor their amount of respiration. And so Hans Krebs had been working with a guy named Otto Warburg, who developed the Warburg manometer, and he is using this to now begin figuring out what the next step is. And this is very much genius work. I don't know if Krebs won the Nobel Prize, but he should have. In that what we're going to do here is start thinking about how these molecules, the carbon molecules, are being released. And the initial thought would be it's just going to be a straight shot, just the way glycolysis is. You're going to just break it down, step, 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 lose the carbons, we're done. It's not that way, and that Krebs was able to figure this out was pretty damn impressive. So let's go into the mitochondria. We're leaving the cytoplasm and heading into the organelle that is known as the warp core of the cell, the mitochondria. And just looking at my board here, sure, we'll stay with this, just to keep these in mind. So we've got the 2 carbons of acetyl-CoA have made their way into the mitochondria. Just a little point, kind of a preview. The mitochondria looks a lot like a bean. I think about this, the cell was a beach ball, about that big. Mitochondria would be the size of one of those foamy circus peanuts, bigger than a jelly bean, about that big. So they're fairly large. They've got 2 main compartments. There's an outer membrane, and then there is a really enfolded inner membrane. And the Krebs cycle is going on in the inner membrane. But that's just sort of where we're heading. Now, the acetyl-CoA molecule, 2 carbon acetyl-CoA, acetyl-CoA. It goes into a system that we call, of course, the Krebs cycle, named after our friend Hans. It's also known, I think not Krebs probably piss people off. It's also known as the tricarboxylic acid cycle, also known as the citric acid cycle. And so it is sometimes abbreviated as the TCA cycle, which stands for tricarboxylic acid. Regardless, I like Krebs, man. So 2 carbons, acetyl-CoA. It is going to be hooked onto a 4-carbon molecule. And the resulting molecule is the 6-carbon citric acid. Now, what we're going to do is start working our way around this organic acid merry-go-round and seeing what happens. The next step is we have a carbon removed as CO2. Just to keep track of this, we had the 3-carbon pyruvate go in. We lost 1 carbon as CO2 when we went from pyruvate to the acetyl group. We now have carbon 2 being popped off as the 6-carbon citric acid. And at various times, yes, I have memorized the Krebs cycle to the note that I can give you the names and stuff off the top of my head, but I don't have it off the top of my head, and you don't need to know it that way either, but it is isocitrate and then alpha-ketoglutarate, not that anybody cares. So we have a 5-carbon molecule. Every time we're going to pop a carbon off, we're liberating an electron, and so we have to have an electron carrier come in. In fact, we missed one here. We had an NAD get reduced to NADH when we lost the first carbon. We've got another NAD picking up electron here. The 5-carbon is going to do the same kind of thing, losing a carbon as carbon dioxide. And we've got another NAD picking up an electron. All right, if you're counting your carbons, and I do, we are now back to the fourth carbon, or to 4-carbon molecule. We have kicked out 3 carbons as CO2. That means that all the glucose that you ate, you now have kicked it out as carbon dioxide. Take one more exhalation. So if I was to feed you, not that I ever would, radioactive carbon-labeled pixie sticks, within 15 minutes or so, you'd be exhaling radioactive carbon. And they actually have done this to little rabbits and things. And so we know this is where the carbon's going. All right. Now, the next step is really just to rearrange the bonds in this 4-carbon molecule to get back to our acceptor. But there's a little bit of energy left in here. We have a couple of things happening. We've got the production of a nucleotide triphosphate, that is, a GDP is being phosphorylated to a GTP. And we still got 4 carbons. We've got now this oddball electron carrier, FAD, comes in, picks up an electron. It is reduced to FADH2, and pretty much that is where we end up recovering and regenerating this 4-carbon acceptor molecule. So, if we now go to our tote board and think about what we're doing with the Krebs cycle, what goes in is, and so here's where, pardon me, I would say what goes in is pyruvate, 3 carbons. And what comes out is 3 carbons from carbon dioxide. That's not quite as official, but it does make the story simpler. However you like to do it, it's fine with me. What goes in specifically to the Krebs cycle is the 2-carbon acetyl-CoA. And we can count them up, but let me just say, how about what goes in is lots of NAD and FAD. Well, 1 FAD. But the main point of this is you're going to need a lot of oxidized electron carriers. And what comes out is going to be 2 carbon dioxides and lots of NADH and FADH2 and 1 nucleotide triphosphate, a GTP. We're going to throw that into the tote board as an ATP because that's just how we do. Now, this reaction per glucose happened twice. So what we can do is go back to our tote board, which we're going to put over here now, and see where we stand in terms of our ATP generating process. So, glycolysis gave us 2 ATP. Mr. Krebs, he's donating a GTP here and then a GTP in the neighboring reaction, the identical reaction. So we can call that 2 nucleotide triphosphates. Remember, we were promised 36 ATP. We still got a long way to go. What the heck? The last step is really and truly the Nobel Prize winning step that I'm going to go through it really quickly and then we'll go back to some of the figures and stuff. It kind of gets into the next chapter. But the next step is the big ATP generating process. This is the process in the mitochondria by which we can make ATP in a really big way. All right, so, part of this is that you have to get a little bit into it to get comfortable with it, and then we can begin taking it apart and seeing all kinds of ways you can poison people and all sorts of interesting events that are going on. but you have to have sort of the first flush through it. And just as a little point, the different acids that you're producing in the Krebs cycle, you make citric acid. There's a variety of things, but my favorites are citric acid, formic acid, fumaric acid, and malic acid. These are all the best acids found in sweet tarts, Jolly Ranchers. All of our various sour candies are using mostly either fumaric or malic acid. So next time you've got a bag of sweet tarts, read what the acid is and think of me. We actually did a little experiment in the food class last year. Our very last experiment was making synthetic pixie sticks, working our way around the Krebs cycle. We've got tons of citric acid. We've always had that around. We have fumaric and malic, which are really good tasting. I bought some alpha-ketoglutarate acid, and no wonder they don't use it. It doesn't taste all that good. It's just not a clean, sharp taste. But if you're ever up in the lab and you want to try some, hit me up. I'll set you up. Fumaric and malic taste really like sour, good. It mixes it with a little bit of glucose. You make a great in vitro lemonade. Add a little bit of vodka, and you're $1 million. All right, so the next step then is to pull the energy out of the rest of these molecules. And if we look at this, we've made a little bit of nucleotide triphosphate so far, but really just a piddly-ass bit. 2 ATP from glycolysis, 2 from Krebs cycle. We got, at least by my calculations, 32 more to go. And that is where this last step comes in. And it is known as, by a couple of names, the electron transport chain. And it's also known as oxidative phosphorylation. Now, this again is a terrible misnomer. Oxygen plays a role in it, but it ain't nearly as critical as this. We should call it the big proton squishy ATP kablooey or something like this. I haven't really come up with my best name for it. ADP rephosphorylation, though, I'm solid on. Oxidative phosphorylation is the term that's used. Part of what we're talking about here is guys were really from like the 1930s up until 1978 or so. Oh, probably 73. We're knocking around trying to figure out what was going on. There was a lot of phenomenon that was observed, and oxygen was clearly involved, but nobody really knew exactly what was happening, and so they're naming it without really knowing what's going on. All right, so in order to talk about oxidative phosphorylation, it's easier rather than to draw it as a pathway and to think about what goes out and what comes in. Let's start with first what's happening, what its job is. And its job can be summarized thusly. Electron transport chain, number one job, it's going to oxidize NADH back to NAD and FADH2 back to FAD, which is really critical because we got to regenerate these guys because we need lots of them for a Krebs cycle. Now, one of the key points here is this. The final electron acceptor is oxygen. That's why you need oxygen, and in fact, that is the only reason you need to breathe. If it weren't for this, you have no need to breathe oxygen at all. You don't use oxygen in any metabolic biosynthesis. You don't use oxygen for anything but the final electron acceptor for reoxidizing these electron acceptors. That's it. And just to make note, the reaction would look more or less like this. We have the electron. We're going to actually add an electron to oxygen, we're going to reduce oxygen. And the reduction ends up looking like this. We make a molecule of water, and we have half of a diatomic oxygen left over. As soon as the next electron comes down the pipe, it gets slapped on there. So the diatomic oxygen half, that is an oxygen radical. This is the active ingredient in hydrogen peroxide, which, where's my bottle of whatever this stuff is? Hydrogen peroxide, Oxivir. Hydrogen peroxide is very, very reactive. That oxygen radical is fairly corrosive. And if you've ever played around with hydrogen peroxide, it can bleach your hair. We're constantly generating a small amount of oxygen radical all the time. One of the fallouts of this is your mitochondria start to get a little bit crispy after a while. And so your mitochondria actually have to be renewed. They actually can divide because they are not able to withstand this oxidative stress all the time. It's another thought that is a bit more complicated, and this is part of it though, is that a calorie reduction diet actually has been linked to longevity. You might be able to extend your life by 15-20% by dramatically reducing the amount of calories. And in part, what you're doing is reducing the amount of oxygen radical that you're generating. Now, this is a double-sided story here, is that you want to reduce the amount of oxygen radical as much as possible. One way to do that would be to not breathe and not eat. But of course, that has some unintended consequences of you're dead. If you can bring your calories down to 800 calories a day, that is the point where you can actually carry out life extension. The problem with that is 800 calories a day is damn slim. You got to make sure that everything you shove into your gob is filled with protein and green stuff. So you're pretty much living on arugula and cottage cheese. And you may live 20% longer, but I think you'll be pissed off most of that time. And so really, what's the point? Where's my burger? All right. Now, that's the first job that the electron transport chain does. Reoxidizing NADH and FADH2, oxygen is the final electron acceptor. The second, now big job, is this. We're going to allow the mitochondria to generate a hydrogen ion, i.e. a proton gradient. And this is why we had to talk about membrane transport before we got into this. The mitochondrial membrane, specifically where this is happening, is impermeable to hydrogen ions, and consequently we build a dam where we got a lot of hydrogen on one side, not much on the other side, and we allow hydrogen to move back along its gradient, and that generates and drives the ADP rephosphorylation. So we're going to generate a hydrogen ion gradient, and this is providing the energy that drives the rephosphorylation of ADP back to ATP. And again, I said it, I stand by it. It is not ATP synthesis. You can kiss my beep. This isn't made for children. It is ADP rephosphorylation. Sorry. This is driving it. All right. Now, the pathway itself is much better as a diagram rather than actually trying to draw the pathway out. And it looks like this. So the whole key to mitochondrial operation is compartments. We got to have a spot where we can put protons out and accumulate them and build a proton gradient. And just another little nerd point. I'm filled with nerd points on this because this is actually a fairly nerdy set of operations. And how many of you ever had this biochemical pathway like in high school? Maybe? I know I had it, and I am sure that my dear instructor in high school had not a clue what she was talking about. But that's okay. So I've actually thought about this for a while. And this next step then is a bit odd. Oh, here's another point. We talk about we're going to make a proton gradient, and it's hydrogen ions. What the hell? Okay, remember when we talked way back at the very beginning of the semester, we were talking about the idea of isotopes, and that isotopes, by my definition, but nobody listens to me, my definition is an isotope means that the atomic nucleus doesn't have the same number of protons as it does neutrons. And I like that definition, but nobody asked me. The definition that is used more commonly is the number of protons is defining the element. The number of neutrons that is most commonly found in the Earth's crust is defined as the element, and anything out of that is going to be an isotope. Turns out with hydrogen, the most common form of it is hydrogen with just a proton and no neutron. And then a hydrogen atom that's got one proton and one neutron, which I would call the element, is the isotope deuterium. And you can put 3 neutrons and you make tritium. And so probably if we started out rather with hydrogen, but with helium, we wouldn't be having this conversation. But regardless, the hydrogen ion that is most commonly found on the planet is in fact just a single proton. So that's why it's referred to as a proton. Fine. A nerd point. Okay. So now the mitochondria looks like that. A Chipotle burrito wrapped up in foil. Actually not a bad example now that I think about it. Inside, it is absolutely packed with membrane. And I draw it like this because to draw it as packed as it is, you'd never really be able to see what's going on. Another one of the ideas here, has anybody ever had this dessert where you can buy basically the Oreo cookie, but it is about that big. It looks like the size of a Petri dish. No filling. It's just the Oreo chocolate cookie. And you fill that with whipped cream, and you make this stack. And then people call it a zebra cake. And if you cut it just like that, that's kind of lame. But if you cut it on the bias, you end up with all these stripes embedded in white cream. That's exactly what a mitochondria looks like, sliced in half, looked at under an electron microscope. But we'll look at some pictures in a moment. Regardless, it is packed with membrane. And just to throw in the names here, a little bit of mitochondrial anatomy, we've got some rather clever names. This is known as the outer membrane. You'll never guess what this membrane's called. The inner membrane. And we've got the intermembrane space. Deep inside, this is known as the matrix. So we're going to do a little bit of blackboard animation and zoom in on a portion of the mitochondria like so. Let's just go ahead and zoom in right here. All right, so taking our magic microscope and zooming in. We got this. Here is the outer membrane, and it's a lipid bilayer. It's got actually a slightly different decorating groups than your standard plasma membrane. I don't think anybody knows why, but it does. Here is your inner membrane, that big hump thing. All right. It's also a phospholipid bilayer. And in here, this is the matrix. This is where the Krebs cycle's happening. All right. There are a collection of proteins and very complex organic molecules, things like various vitamins like B12 and If you ever heard of something called CoQ10, it's in here as well. Again, as a supplement, completely a waste, but whatever. And they're set up like so. So this set of complexes of proteins and vitamins, they are just jam-packed all throughout the matrix. That's what the matrix is doing, is providing a ton of surface area for this next step. We've got a protein complex that is a transmembrane protein, and it is known in the biz as complex 1. Then we've got complex 2. And you'll never guess what this one's called. Complex 3. Lastly, we have complex 4. Then over here we have something that doesn't look at all like the other guys. It is what the book would call ATP synthase. Yeah, and I wrote it up, but I refuse to call it that. It is the ADP rephosphorylator. It's also known in the biz as, yes, complex 5. If you want to memorize your complex names, go ahead, but we don't need to. Here's what happens. Remember, their number one job is to reoxidize NADH and FADH2. That means NADH backs up, dumps its electron onto complex 1. Beep, 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 dump the electron. And in fact, complex 1 is known as NADH oxygenase, I think, regardless. Or NADH dehydrogenase. NAD has been regenerated. It's given a little pat and told, go back out and give me more electrons, boy, and it runs out like a faithful dog back out into the game. Complex 1 is going to hand the electron over to complex 3. We'll get to complex 2 in a bit, but complex 2 is kind of lame. Complex 3 takes the electron and hands it over to complex 4. Complex 4 takes the electron and hands it off to oxygen. And that is where we now make a molecule of water and the half diatomic oxygen molecule. And actually, that is the end of the game. Elvis has left the building. The carbon that we brought in is out, and most of the electrons have now been dumped onto oxygen to generate water. Let's just go ahead and fit in the weak sister of the mix, and that is complex 2. Complex 2 is responsible for taking FADH2 and oxidizing it to FAD. The electron is then going over here, waiting its turn and making its way down. So now, at this game, we really have done and gotten rid of everything. The last step, and this actually truly was the last step to figure out, this is the Nobel Prize winning point. Complex 1 can take a hydrogen ion and pump it out. Complex 3 can take a hydrogen ion and pump it out. Complex 4 takes a hydrogen and pumps it out. Complex 2, don't do jack. Again, in my super soldier army, I'm going to make complex 2 pump out a proton. That would give you just a little bit better energy production. Now, the membrane here is completely impermeable to hydrogen ions. The membrane out here actually can allow hydrogen ions to pass back and forth. By carrying out this hydrogen ion pumping, we generate a pH difference. We got a pH of 7 here, and same in the cytoplasm. In the matrix, we have a pH of, wait for it, 8. And you might say, oh, Big deal. But remember, the pH is a logarithmic scale. We've got 10 times more hydrogen out here than we do in here. 10 times. That's a pretty big gradient. So the only place that hydrogen ions can get back through is through ATP synthase. The hydrogen ions take their turn, they zoom back in, and this is what drives the rephosphorylation. ADP plus the inorganic phosphate generates ATP. Yay! And if we do our ATP count, what we got then, glycolysis 2 ATP, Krebs 2 GTP, electron transport chain is the rest. So what? 32 ATP. Pretty important, I would say. All right, now what we have outlined here is actually all kinds of fun biochemical hacks you can do. Different and interesting ways to kill people, some fascinating diet drug tips, all kinds of metabolic poisons. Lots of things fall from this, not the least of which is what happens if you were to shut off the oxygen. We'll pick up on that on Wednesday. And we should all be here. We should maybe meet in Albert Taylor Theater because this is one of my, I think, more of my more engaging lectures. I always flatter myself, but what the hell? Have a good day.
Meet out on the quad.
Mm-hmm.